Okay, so let's talk about the exam 3 lecture 2. We will talk about the microorganism metabolism, the energy transfer. So very important in this section, we need to know how to calculate the whole energy, the total number of the energy, the ATP's generation during complete aerobic respiration, and some of the other concepts. Now, also, when I finish this Zoom video, you have to look at the video what I put on the YouTube channel, which is aerobic respiration, and the one section of the lab 9 and the lab 10 when I wrote on the blackboard, which is about the fermentation and electron transport chain. So first, this is the nutritional types of organisms. So based on the nutritional types of the organisms, the sources of carbon, energy, and electrons, we could be categorized bacteria into autotrophs and heterotrophs. So autotrophs is using carbon dioxide as the only biosynthesis carbon source. Now heterotrophs is used as others. Most of them is molecules from other organisms. Now, based on the energy source, we could categorize bacteria using light as an energy source. We call it the phototrophs. And we could also we could say most of the bacteria in our lab, which is using organic or inorganic compounds, so we say it's chemotrophs. Now, based on the electron sources, we have some of the bacteria using reduced inorganic molecules, we call it lysotrophs, and others use organic molecules, we call it organotrophs. Now, you need to know all the microorganisms, they actually is a good combination of all those three resources. Therefore, you get this table. Okay, it's a very, looks like it's a little bit complicated, but it's a combination of everything. So I want to just to briefly mention something. When we talk about in the lab, all of the bacteria, what we are talking right now, is chemoorganoheterotrophs, which means organic carbon coming from sugar is a carbon resource. Organic chemical, also from a sugar, is the same as carbon resource, is the energy resource. Electron resource, including donor and acceptor, also the same as carbon source. So chemoorganoheterotrophs, their carbon source, energy source, and electron source are exactly the same. Therefore, most non-photosynthetic microbes, including most of the pathogens, fungi, and proteus and archaeas, those belonging to chemoautotrophs. So we are talking about this right here. We used to have some section talk about photolysoautotrophs and the photoorganoheterotrophs, but we just skip this semester because we have a limited time. But I want to let you know, these are really important bacteria. It does not mean they're not important. For example, cyanobacteria, they contribute to almost most of the 80% of the oxygen generated in the world. And you see those sulfur bacteria, which use sulfide as energy. Those are anaerobic bacteria. Where you can see those in a Yellowstone National Park. Okay, now when we talk about the chemolysoautotrophs, these bacteria usually is using other energy resource, not rely on glucose. They use sulfide, hydrogen oxidized, methanase, nitrogen fixing, oxidizing. They contribute a lot to the environment, especially nitrofuran bacteria. We will talk about nitrogen fixing, nitrogen cycle, all those things, and also hydrogen mesogens, which generate methyl in the environment. So those are not really belonging to this class. That's belonging to water microbiology or environmental microbiology. So you have other classes. We'll be talking more specific, all these different types of the microorganism. So I'll just let you know what we are doing right now is only one category, is chemoorganoheterotrophs. So this is what we're going to talk. Chemoheterotrophs, we're going to talk about aerobic respiration anaerobic respiration, and the fermentation. And remember, I already have a video for that, so we go over the slides right now. This is what I draw on the blackboard already. So if electron donor is an organic chemical, and the electron acceptor is an inorganic chemical, and that inorganic chemical is oxygen, that is called aerobic respiration. In other words, if that inorganic chemical is other inorganic chemicals besides or other than oxygen, it's not oxygen, then we call it anaerobic respiration. And the ATP generation mainly through oxidative phosphorylation. And we mentioned in the video the difference between oxidative phosphorylation and the substrate level phosphorylation, which is related to which one has electron transport chain incorporated. Oxidative phosphorylation has electron transmitted chain in their substrate level phosphorylation without electron transport chain. This is a major difference. Okay, so we're going to talk about use endogenesis electron acceptor. So most of them, they will have an intermediate chemical. We call it a metabolic precursor, actually, play an important role for all the pathway. Now, which one is more important? Is pyruvate. Now, where is this pyruvate comes from? This pyruvate, which comes from glycolysis, okay? So this is the bottom line. So we talk about the fermentation, you need to know. There is an intermediate products, very important. It's called the pyruvate. That pyruvate is generated through glycolysis. Now, you also should know, fermentation, they generate ATP. How many? Only 2 ATPs. They through substrate level phosphorylation. They do not encoding electron transport chain, which means they not rely on protein motive force. And I mentioned what is protein motive force before, which means all the hydrogen is accumulated inside of the cell membrane, then can be repelled out of the cell membrane, and that's a potential energy will later on trigger the ATP synthase, generate ATP, and coupled by flagellar rotation. And we will mention real quick in the next few slides. So this is a 2 different type of the fermentation. We mentioned pyruvate reduced to lactic acid, NADH oxidized to NAD. This is called homo fermentation. Pyruvate all the way reduced to ethanol and coupled with NADH, oxidized become NAD plus and have an intermediate product called acetaldehyde. This is called heterofermentation. And remember that NAD plus is very important, which means this electron carrier is recycled, can be used for another glycolysis coupled with fermentation process. Okay, so that's important. So this is the fermentation. Oxidation NADH produced by glycolysis, pyruvate electron acceptor, substrate only partially oxidized, no oxygen involved. They will be ATP formed only by substrate level phosphorylation. So you can see the NAD. They contribute to go there to the glycolysis. This is glycolysis. Remember, I draw on the blackboard in the video. This NAD is recycled. This NAD must be recycled, otherwise it will be died. Okay, so that is called the glycolysis and the fermentation pathway. They are coupled. This is the application for the fermentation. You like Swiss cheese? Because probionic bacteria, they can generate carbon dioxide and acid taste. You like cheddar cheese, yogurt, soy sauce, that's lactic acid bacteria. Do you like wine? We have a Saccharomyces, that's called beer wine, that's called beer yeast. They generate ethanol and carbon dioxide, have beer and wine. Now, or the ladies like the nail polish remover, that is actually coming from Clostridium, which is the main products is acetone and isopropanol. You can get that one bottle from Ultra or from Sephora. That is also coming from fermentation. Now, last one, the vinegar. That generates acetic acid through fermentation, mainly by E. coli and Acidobacter. Remember, this E. coli is not a pathogen. That's E. coli 9037, which is a beneficial bacteria. Now we're going to talk about aerobic respiration. We talk about glycolysis, TCA cycle, electron transport chain, and they will generate ATP. So we already talk about those. We're going to be a little bit easy. Okay, this is called Embden-Meyerhof Pathway, is another name for glycolysis. That happened for most of the bacteria, which is in cytoplasmic matrix. But specifically for mitochondria, for the fungi and the yeast, molds, those are in the mitochondria. There is two steps of the aerobic respiration. This process is in the absence of oxygen, but functional in the presence absence of oxygen. There is a two-phase, 6-carbon phase and 3-carbon phase. Now I'm going to skip this one because I already talked about on the blackboard, okay, how many we generate. Mainly, we're not going to talk about this enzyme. Mainly important, at the 6th phase, there is 2 ATP has been used. And at the 3 phase, there is a 4 ATP generated through substrate level phosphorylation. But don't forget, when we talk about only 2 ATPs generated, we not included this NAD reduced to NADH2 because we think it's a closed cycle. But when we talk about aerobic respiration, this will be included. NAD reduced to NADH actually involved in an electron transport chain, so they generate 3 ATPs here. And also remember, here you have to be double because there is 2 3-phase chains. Okay, this is the final formula of the glycolysis, and we already mentioned the glucose we're going to generate 2 pyruvate, have 2 ATPs. Now remember, this is a closed cycle, okay, not related to the in the absence of oxygen. So we're going to talk a little bit more. Oxidative phosphorylation, which means related to electron transport chain, ATP synthesis, and the associated respiration. Now, this is what I draw on the blackboard lots of times, the electron transport chain. From NAD, FM, FESFAD, cytochroma Q, COQ, cytochroma group, and the cytochroma C, A3, and oxygen. And where are they? In the eukaryotes, usually it's in the mitochondria, so they generate protein motive force triggers the electron transfer, and they transfer always from higher energy to lower energy, and always from the more negative voltage values through 2-half reaction to a more positive value based on 2-half reaction, because more negative, which indicates it's a more good electron donor, and a more positive value, which indicates it's an electron acceptor. So this is a very important, how we really generate ATPs. This is a theory called Chemiosmosis. This generated by a British scientist called Peter Mitchell in 1937. In 1950, this is a Nobel Prize award results. So what's the story? When you see the electron transfer, they actually activate called dehydrogenase. What means dehydrogenase? Which means they're going to release hydrogen compounds, and that called protons. They will be repelled outside of the cell membrane, then accumulate there, then generate this potential energy called proton motive force. Then this potential energy will be trigger ATP synthesis. And at the same time, when there is so many H protons outside, they're also going to be come back. And once the ATP synthesis has been activated, the ADP will become ATP. Then what happens is these hydrogen compounds will go inside through active transport. Remember, this is a channel, which is a carrier, and then they also need energy, which is ATP. But for bacteria, more important, they generate energy, which means they're going to have to moving. How they move? They're going through rotation of the flagella. That's a real movement. Okay, so overall, which means there is accumulation of the protons outside of the cell membrane, triggered by hydrogen dehydrogenase. Okay, then generate accumulated potential energy called the proton motive force, then activate ATP synthesis, generate ATP. At this moment, because of too many of the proton compounds outside, so they coming back and coupled with rotation of the flagella. That is called chemosmosis. So this is what happened from proton motive force. We are talking about right now is this way. Electron donor, electron transporter, activate proton motive force, generate ATP, active transport, and bacterial flagella rotation. This is a photobacteria, that is through the light, have a photosynthesis. A good example is cyanobacteria. At the end of the day, they also generate proton motive force. So we're not going to talk about this section. We're only going to focus on is right here. This is the transition step of the pyruvic acids. So pyruvic acids, decarboxylation become acetyl and have the coenzyme M become acetyl-CoA. This is the transition step of followed with TCA cycle, okay? This is the TCA cycle. I already talked about a lot on the blackboard, so you can write here. More importantly, you're going to see whenever you see NAD reduced to NADH2, that means there is a 3 ATPs generated. That is going through oxidative phosphorylation. And when you see FAD reduced to FADH2, what happened is also oxidative phosphorylation. 2 ATPs generated. And you don't forget there is a substrate level phosphorylation right here, which is GTP becomes GDP and all ATP becomes ADP becomes ATP. Okay, we already drawn the blackboard, so I'm going to skip that part. This is the calculation, the 38 ATPs. And remember, we still use the older calculation, but you can use a new one. That's okay. The new one, which is taking consideration of the phosphate to oxygen ratio, so 2.5 ATP going through NADH, 1.5 ATP going through FADH instead of 2, and here instead of 3. So you can do the calculation by yourself, but you can use the older one or the new one. I basically use the older one. This is energy aerobic respiration, how the energy going to calculate it, and I already write on the blackboard. This is some reference for you. And very quick, we talk about anaerobic respiration. We will do one test in the lab. So first of all, anaerobic respiration is using other inorganic chemical as an electron acceptor other than oxygen. So anaerobic respiration generates less energy compared to the aerobic respiration. Why? Because the distance from the beginning of the electron carrier to the end of the electron acceptor, the distance, the voltage difference is less or much less than aerobic respiration. So they generate less energy. Now, these are all the examples of those anaerobic respiration. Lots of them are environmental bacteria. Methogenesis, desulfur could be desulfide, and these Pseudobacillus geobacter could generate ferric acid. And these anaeromonas, these are all environmental bacteria, so we just skip that. This table is important. It gives you a conclusion of the energy generated through Chemoorganoheterotrophs. So you can see how many ATPs they're going through the aerobic respiration, 38. How many is through oxidative phosphorylation, 34. And how many is through substrate level phosphorylation, which is 4. Anaerobic respiration, 36 or less ATP. 32 going through oxidative phosphorylation, 4 going through substrate level phosphorylation. Now I want to mention the anaerobic respiration also has a TCA cycle, but that is called a modified TCA cycle, related to each specific bacteria. So we don't mention that in here, but I do want you to learn TCA cycle for aerobic respiration. Now fermentation, only 2 ATP generate, coupled with glycolysis. So we already mentioned. Now, very important, you have to remember this table, and also very important, when the exam questions, go to see this table as a reference and look at my video when I put on the YouTube to do the calculation. That's a very detailed, we explain the calculation. Now, last one I want to mention, this is called the Pentose phosphate. Pentose phosphate pathway. we're not going to go very detailed, but you need to know the big difference is that instead of they generate NADH, they generate NADPH at a reduced energy, and they use for biosynthesis. So this is actually related to the catabolism. It's actually related to anabolism instead of catabolism. So what is the conclusion for the pentose phosphate? They will be generate 5-carbon sugars, and further they will biosynthesis NAD, DNA, and RNA. Their reducing power is NADPH, and they also called precursor metabolites, and those precursor metabolites will become synthesis DNA and RNA. So pentose phosphate is an anabolism process. Okay, we just be very briefly right here. I used to have a question for the exam, but this year we're probably not going to do it. But I just let you know, I want to skip this one. You're probably going to learn a lot in your biochemistry class, because for bacteria in their very short time, we don't really care about how they biosynthesize DNA and RNA. If you go taking further class, which is microbiology, uh, um, metabolic specific metabolism classes for microbiology major, they may go through so detail, but in here for general microbiology, we spend very less time talking about the pentose phosphate. You only need to know is all these conclusions. Okay, so this here we ended this part, which is metabolic pathway. Remember, more important is look at my slides and the video recording in the lab section, lab 910, typically lab 10, and the aerobic respiration calculation, which is I put on the put on the YouTube channel. Okay, thank you.